Hello and welcome, dear students. How are you all? Please take your seats, dear students. Yeah. Students, please take your seats. Okay. So, all right. Yeah. Students, please report your attendance, and we are beginning right away. Yeah. Please settle down. I hope you had your lunch. Good afternoon, everyone. All right. Yeah. And know that your people are taking your time. It's okay, but let me start now. All right. Senapati and yeah, good afternoon, Senapati students, and good afternoon, Shillong students. I would ask you that before we begin, just please quickly someone send your attendance, both Shillong and Senapati centers. Yeah. The other centers are not online right now, so they can, when they join, they can. So please report your attendance first of all. Please do that quick. Okay. All right. Okay, Senapati, thank you. And Shillong, you're the only one remaining. I can see you people there, but just count the number of students and just report it quickly. All right. Okay. So now let's begin. In the last class, what I had asked you was I had begun a new chapter. All right. So let us start from where we left in the last class. I had started transport and mineral nutrition plants. Basically, we have to discuss how does transport occur in plants. Okay. So basically, we have to discuss the means of transport in plants. Transport in plants. Okay. So in this chapter, we had discussed that the only way, numerous ways, in which substances can be moved from one places to the other place is by osmosis or by diffusion. Diffusion is of many types: simple diffusion or facilitated diffusion. And there is another method called active transport also. Then there is osmosis. Okay. Osmosis is particularly only for water, not for anything else. In plants, when you talk about osmosis, it is only for water, only for transporting water. For transporting ions such as sodium, potassium, you will use active transport. For essential things like glucose, anything that is essential for the survival of cell, you will use active transport. For the other things which have a concentration gradient, we do not need to spend energy. We will use facilitated diffusion. Okay. So there are some methods I told you in the last class. Okay. I had also sent you a PPT. So let's play it quickly. I had asked you to take a look at this PPT that I had given you. So, all right. When we talk about transporting substances, we have to cross a cell membrane. The cell membrane has the function of protecting the cell, controlling the incoming outgoing substances. It has the function of maintaining ion concentrations of various substances, right? It is selectively permeable and allows some molecules in, others are kept out. And this maintains the balance of the cell, and this balance maintenance is called homeostasis. All right. Okay. This cell membrane has the nature of phospholipid bilayer. Okay, it is a bilayer of phospholipids. This is how it looks like. The structure of plasma membrane is explained by fluid mosaic model, and this model was given by Singer and Nicholson. This model I have already explained to you when I taught you the cell chapter, and I told you that the proteins in the cell membrane, they play a very, very important function. They help in transporting substances from one side to the other side. So this part I had given you short introduction when I taught you cell chapter regarding the plasma membrane. Moving on, so we have some methods of moving substances: diffusion, osmosis, facilitated diffusion, active transport, okay, and the model shows the movement, how the things are going to move, okay. So they love water and dissolve substances which are hydrophilic, okay, but only lipid soluble substances can move freely. Okay, I am not able to play the animation, just wait a second, I will play some things for you. And then there is cellular transport, passive transport, active transport, cell does not use energy, and so this, right. So these are the things: diffusion, movement of molecules from an area of high concentration to low concentration. We have done all this. So first of all, when we begin today, let us take some notes, and after that we will discuss how we have to proceed further, right. All right, so let's begin. Please put the heading transport in plants. Okay. So when we talk about transport in plants, transport of substances, transport of substances in and across, sorry, across, not inside always, and that's not true, across the cell membrane is essential for the survival of cell, right. And cell membrane plays an important role. Cell membrane is just the plasma membrane. The plasma membrane present around the cell is called cell membrane. If it is present around chloroplast, we will call it chloroplast membrane. Or plasma membrane plays an essential role in allowing substances across it. So we will have to study the nature of this membrane also, substances through it. Okay. Next we will study the means of transport, right. So let's take a look at the means of transport. Okay. One more thing, students, have you solved the questions? I wanted to ask you the questions of cell chapter from module, cell division, from module, any guide, anything. Have you solved these questions? At least from the module I expect that you people must be solving. Have you done it, yes or no? Should I think I should maybe give you some questions first. If you are not solving by yourself, I am not sure how many people are following up, because I keep on following the tests that you are giving, and I know which people are performing well. So yeah, you can discuss your issues with me, like if you are lagging behind in some topic specifically, please let me know. Okay. If you are lagging behind in some particular chapter, something is not clear to you, please let me know. So up till now, whatever we have covered, okay, you will need notes for it, you will need to study it for it, and you will have to solve a lot of questions. So because I'm not sure how many people are solving by themselves, so I think, do you want me to give questions from the beginning, or do you want me to give questions from cell and cell division, the last chapter only? Yeah. If you want, I can give you from the beginning. You tried some. Okay, it's very good that you tried. I think it's time that I give you more questions, more than the module questions, so that you can solve it. So I'll give you questions according to difficulty level. Okay, so tell me comfortably, how much time are you devoting to biology per day? So I can give you approximately 50 or 100 questions based on this information. How much time can you devote for biology per day? No idea, no time at all. You must be giving some time. Okay, so give me an estimate that you are giving 1 hour, 2 hour, something like this. So by that estimate, just an average, ask among yourselves. So are you giving 1 or 2 hours at least to biology per day? Okay, so all right, no answers. Give me some answer. Okay, 20 minutes, 30 minutes, something, some value. All right, nobody's answering still. Okay, I guess you are giving very less time. Okay, so look, my course is progressing at a steady rate. Okay, some people are giving 2 hours. I'm very happy that you are giving at least 2 hours. It is very necessary that you give at least 2 hours for biology. And look, botany is a different thing and zoology is a different thing, but both are essential if you want to pass the NEET examination. So I understand that my topics might feel a bit different for you, but I think I should now give you lots of practice questions so that you become very adept in solving them. Okay. So I saw your performance in your test papers. So some people were doing biological classification chapters very well, and some people have not attempted questions related to some few topics. So I saw this gap in them. So some people are not studying, or maybe there is some issue on their part. So I think, look, what you can do is, you can personally message me on WhatsApp. That's this is my problem. I would feel it better if you message me on the group, but it's okay. If you have any particular problem, you are not feeling very calm about it, and you're not happy about your performance also, just message me, sir, I am having this problem. I'm not able to do these topics well. I'm not able to understand this concept. So what can you do to help me out? Okay. So if there is any issue related to studies in general, you can also ask me about it. So I'll give you information based on what you are doing. I will ask you some questions. So based on that information, I'll give you an advice that is going to be different for everyone. Okay. So try to engage with me a bit. Let me know your problems, and I'll give you personal advice based on your performance. I'll see your name in the tests also. I'll see what you are doing, because I will ask you some questions as well. Okay, not related to topic necessarily. Okay, do not worry about that. That I will ask you some questions related to topic. Yeah, that's a different thing. I want to ask you questions related to that. I want to ask you some things about your studies first. Then I'll give you an advice how you should prepare for your exams. Okay, so feel free to reach me out. Okay, I will look forward to your messages, and then I'll decide how to consult you, how to give you a better advice so that you can do better in your tests. Okay, so in the next test, please try to attempt questions. A lot of questions, a lot of people are doing very well. Okay, so I'm not taking any names here, but I know there are 3 students at least I'm very happy about. They are doing very well. The top 3 ones, they are doing very well. But the other, please, at least there are 10 students, I can say that with confidence that you people have the capability to really pass the examination, and you'll get a seat in the medical college, right? Yeah, just a second. So the thing is, for the others, I must give you this advice. At least for biology, please don't feel so much bad about it. Biology is a very interesting topic. If there is something you are feeling bad about, you are feeling like you have left some lectures also, so please reach me out. I'll tell you what to do. Okay. Okay, Shillong student, you have a doubt. Sir, in anaphase 1, there is a statement the number of chromosome is reduced to half. Okay, yeah. In anaphase 1, there is a statement the number of chromosome is reduced to half. Look, the chromosome number is not actually reduced to half still. It is not reduced to half until telophase is there. Chromosome number is not actually reduced to half. The chromosomes have not been split into half. This is a different thing. If there are 46 chromosomes in anaphase 1, the 23 chromosomes will go to one pole, the 23 chromosomes will go to other pole. They have not separated. It is still one nucleus. The nucleus is going to divide in telophase. Actually, the chromosomes have not divided yet, but you can say that when telophase will be finished, the chromosomes will be reduced to half. Okay. And now you are asking me if there is a bivalent chromosome and it has a complete chromosome. The bivalent chromosome is actually, look, bivalent means two chromosomes. Bivalent or tetrad, bivalent is different thing and single chromosome is different thing. A single chromosome has two chromatids. A bivalent has four chromatids. Okay, there is a conceptual problem here. A bivalent is having four chromatids. One chromosome has two chromatids. Each chromatid, when separated from the other chromatid, becomes a new chromosome because it is a duplicate of the other. Okay, there will not be any loss of genetic information if chromatids separate from each other. This is what you need to understand. When DNA synthesis happens in S phase, then a new chromatid is formed and this chromatid is joined to this at centromere, right? This is a chromosome. We can call it a chromosome only. Okay. When it separates from the other chromatid, it will still be a chromosome. The identity does not change. The only thing is, when the cell is going to divide, the chromosome has a double copy of itself joined at the centromere. This DNA, this chromatid, extra chromatid was made during the S phase. Okay, this was made during the S phase. Now it has not split. In anaphase 2, it will be split. Then you can say the chromosome number remains equal. I hope you understand your question now. In anaphase 1, the statement, the number of chromosome is reduced to half. If you want to talk about it really, the thing is, for each nucleus, there will be half number of chromosomes. Each nucleus, there will be half number of chromosomes. Okay. Suppose you are sitting in this class, Shillong students, and I ask that because you are 12 students, so 6 students sit to one side and 6 students sit to one side. Okay. Then I can ask you people to form pairs. Okay. When I ask you people to form pairs, this pair is called a chromosome. If you separate from your pair, you will still be a chromosome. Okay. It's just like a dancing competition. Crossing over means exchanging some parts. You can exchange your watches, you can exchange your pens, you can exchange your hair bands also. Okay, a lot of things you can exchange. So that can be called as crossing over. But when you separate from each other, when everybody goes back home, you are still an individual person. That is the same case with chromosome. When the chromosome has to divide, it makes a clone of itself. The clone is called sister chromatid. The bivalent is not a single chromosome, it is two chromosomes. What about the number of chromosomes? Look, because you are having this doubt, should I play the video that I asked you people to do, watch? What about the number of chromosome? I say this again. The number of chromosome in meiosis 1 is reduced to half. It's you are having a very valid doubt. I really welcome you that you are asking you this question, because it seems very confusing. How is the chromosome number reducing to half and then it remains the same? Somebody who does not know biology, he will say that if chromosome number is reducing to half, then in meiosis 2 it should become 1/4, right? They can say this. because they do not know what is the concept behind it. The concept is we make a copy of the genetic material in S phase. This genetic material copy is called sister chromatid. It will be visible when cell division starts. Before it, it will not be visible. These sister chromatids, it is an exact copy. Okay. Now, when meiosis will happen, meiosis 1, these sister chromatids do not separate in meiosis 1. The homologous chromosomes separate. This is a very beautiful thing that you must understand. What is our chromosome number? Our chromosome number is 23 pair or 46 chromosome. But these 46 chromosomes, they are basically 23 pairs. 23 are from mother, 23 are from father. Another important thing, these 23 pairs, they are exactly like each other. If one chromosome has some length of sister chromatid like say 2 nanometer, the other homologous chromosome will have 2 nanometer only. Another chromosome might have chromosome number 2, second pair, they might have chromatid length 1.9 nanometer. The other pair member will also have 1.95 nanometer. Whatever is the length, it will remain constant. So the thing I want to tell you is there are pairs of chromosomes. When you talk about diploid number of chromosomes, please think of them as pairs. These are basically pairs of chromosomes. So the pairs are going to be unpaired. The pairs are going to be unpaired in meiosis, cell division, and they will be paired again during fertilization. The chromosome number will be reduced to half in meiosis. The pairing is broken in meiosis. Okay. So when I talk about meiosis cell division, the pairing is broken. Each homologous pair of chromosome, they will separate from each other. And when they will be united, they will be united when there is fertilization. The chromosome number will be restored. Look, the gamete does not have any function. The gamete is not a cell which has the function of a skin cell or liver cell. So if it has half number of chromosomes, it does not matter. It does not matter. It will perform any function only and only when it has the function of basically becoming a zygote. After becoming a zygote, then becoming an embryo, then it will show its effect. Okay. So the number of chromosome in meiosis 1, after anaphase 1 comes telophase 1, will be reduced to half. And it will remain half in meiosis 2 also. It will not become 1/4. It will become 1/2 only at end of meiosis 1, and after meiosis 2 also it will remain 1/2. If there is a doubt still, please let me know right now. I can play you the cell division video and we can talk step by step what is happening and why it is happening. Are you a bit sure about your is your doubt solved now, or should I continue further? Okay, I hope your doubt is clear. If there is anything else you want to know, please message me, and yeah, please also mention your name, dear student, what you are asking right now. Just mention me your name so that I can remember. Yeah, please write your name also. I want to remember all your names, okay? So all of you are very dear to me. I understand that you have doubts, so it's okay. Please always tell me your name so that I can remember, and I will reach out to you. Okay. Alright, Shembalang, I'll remember your name. At least I will try to remember your first name. Your second name, if I am not able to understand, I'll ask you when I come over your center. Okay. So Shembalang, I'll remember this doubt that you had. If you have any doubt, please message me. Just write that your name is Shembalang, and I'll get back to you. Okay. Alright, let's continue. So I told you that we were talking about the transport in plants. So first of all, we will discuss what is happening in the cell, across the cell membrane. After this, we will move on to the multi-level transport in plants, in which many cells will be involved. First of all, we will talk transport in plants at cellular level, then we will transport water minerals through xylem and food through phloem, and these will be discussed in stepwise manner. Okay. Means of transport. What are the ways that substances can move from one place to the other? Means of transport. Number one is diffusion. Diffusion. Okay. So what is diffusion and how it happens? Okay. Diffusion is the, please write, diffusion is the movement of substances from their higher concentration to their lower concentration. From their higher concentration to lower concentration. Okay, it is the movement of substances from their higher concentration to lower concentration. Okay. So this movement is non-specific. It can occur for anything. It can be for solute also, solvent also. It is not mentioned here that which substances will move. So I am telling you, if something is dissolved in something, if something is dissolved in something, if you are talking about diffusion, diffusion can occur of anything, but the diffusion of gases is fastest, the diffusion of liquids is intermediate, and the diffusion of solids is least. So please write diffusion can occur for solids, liquids and gases. For solids, liquids and gases. Okay. But diffusion of for gases is fastest, diffusion rate, gases fastest, liquids intermediate, solids it is the least rate. Have you studied Graham's law of diffusion? Graham's law of diffusion. Rate of diffusion is inversely proportional to the molecular mass of the gas. Graham's law of diffusion, the rate of diffusion is proportional to 1 upon under root of m, molecular mass of gas. Yes or no? Okay, you don't remember, or maybe you have not studied it. For example, if I have hydrogen gas is lighter, oxygen gas is heavier, so their rates of diffusion under root 1 upon m. So if I compare a diffusion of hydrogen versus oxygen, so hydrogen will be diffusing 4 times faster than oxygen gas. Okay, so this will come in physical chemistry, don't worry about it. Now, first of all, tell me, have you seen diffusion of gases? Have you seen diffusion of gases? If somebody in your class, suppose some boy enters, a new boy enters your class, and he is having some Axe perfume or deodorant sprayed from head till bottom. Okay. So, yeah, I have met such people, and who spray a lot of perfume. They spray so much perfume that if somebody is enclosed in a lift, for example, one day I have a lift at my apartment, and there are some people who spray so much perfume that when you're confined in a small space like a lift with them, it becomes very hard to breathe. Okay, so yeah, if somebody is having sprayed some perfume and he's sitting at one corner and you're sitting at the other corner of the class, you can smell that, okay, perfume. But if any person is standing in somewhere in between, okay, in the middle of the class, he will smell the perfume faster than you will be able to. Okay, so diffusion is happening for gases at the very fast rate. But suppose if we have something like a liquid, and suppose you are having some apple juice, but and if you're carrying some apple juice in your glass or something, if you want to open it, nobody will know that you are opening apple juice container. Okay, nobody will know that you're opening an apple juice container. But if you take your nose right up till the top of the liquid, you will be able to smell that, yeah, very nice smell of apple juice is coming, or orange juice is coming, something like that. Yeah, orange juice is more fragrant, so let's take orange juice. Okay, so the thing is, if it is a liquid, the diffusion will happen at a slower rate. But if somebody opens a bottle of perfume or becomes a bottle of perfume by spraying a lot of perfume, so in that case you will see it is happening faster. Solids also undergo diffusion. Can you tell me any example of solid diffusion? Can you tell me any example of solid diffusion? Anybody? Can you tell me any example of solid diffusion? Nobody? Okay, all right. Okay, I can see what you people are doing, what the people at the back benches are doing. I can see everything. Shillong students, I am enjoying it also. It's okay. All right. So the thing is, yeah, alloys. Alloys can be made by heating. If you talking about, yeah, very good example. Very good. Whoever has answered the question, making of alloys is an example of solid diffusion. Another way of demonstrating solid diffusion is, if you increase the temperature, you can make an alloy. If you do not increase the temperature, you cannot make an alloy. So alloy is basically solid. We convert them into liquids, and then the diffusion can happen. But if I am talking about making solid diffuse with another solid, this is very difficult. Alloy making normally employs converting metals into liquids, or making them beating them together. A lot of methods are there. Normally the quickest method will be mixing two liquids together, okay, or heating two solids together. So the thing is, if you take a glass, sorry, not a glass, let's take a zinc strip and a copper strip, place them in vacuum for 2 years side by side, they will automatically be welded after 2 years. If you place two metals together, clean surfaces, and place them together, after 2 years they will join together. But the surfaces should be clean, not oxidized. There should be no rust on the surface. So this is how you can demonstrate it, but it will take 2 years, so it's a very long time. So I hope you understand the case of diffusion. Now the diffusion is of two types. Diffusion is of two types. Simple diffusion and facilitated diffusion. So first put the heading simple diffusion. simple diffusion. Okay, simple diffusion. So what is simple diffusion? Simple diffusion is simple diffusion. Okay, simple diffusion means, it means substances moving freely from one place to other, from one place to another, without help from supporting proteins. Okay. Without any help, just write without any help. Without any help is sufficient. Without any help. So substances are moving from one place to another without any help. Also, example, opening a perfume can, or there can be other examples also. Okay. Simple diffusion, for example, we human beings and animals also, they have the capability to form gaseous substances inside their digestive tract and releasing hydrogen sulfide, ammonia, and some volatile fatty acids into the atmosphere. So nobody wants to talk about it, right? So that is also an example of diffusion. So okay, all right, opening a perfume can or any volatile substance, or any volatile substance okay, in a room. So the people can, on the other side, can experience the flow of gases, and they can tell you what you have brought for lunch, what you ate for lunch, or what you are, what perfume you are wearing. A lot of things can be told about us, right? So a lot of things like this can happen. Anyway, let's continue. This is simple diffusion. So this is like this only. Okay. Now let's talk about facilitated diffusion. The next thing, the more important thing, facilitated diffusion. Facilitated diffusion. Okay. So what is facilitated diffusion? What is facilitated diffusion? What is facilitated diffusion? Just a second. Facilitated diffusion, for watching facilitated diffusion, let me show you a short video, and then you will be able to understand it better. High concentration gradient, phospholipid bilayer, everybody recognizes it, and then there is a low concentration gradient, right? And substances are moving across this barrier, tunnel like structure, okay, and after that, this is the basis of facilitated diffusion, right? Okay. Facilitated diffusion. So please write. Okay. Diffusion which happens with the help of a protein carrier. A very good Shillong student who answered, which happens with the help of a protein carrier is called facilitated diffusion. And these proteins are basically integral proteins. Protein carrier is called facilitated diffusion. And facilitated diffusion is also an non-energy intensive process. Non-energy intensive process. That means energy is not wasted in performing facilitated diffusion. Okay. So you can write now. In facilitated diffusion, substances will be transported, facilitated diffusion, substances are transported along their concentration gradient, are transported along their concentration gradient and without the expense of energy, without the expense of energy. Okay, so energy is not expended, and basically this is how it happens. Energy is not expended. Okay, how? Yeah, one more thing. Right, so let me play this video again. I'll explain on the way. Look, you can see that there is a protein channel here, okay, and protein channel is basically like a tunnel, and concentration gradient, this substance which is present on the outside, it cannot cross the membrane by itself. It needs the help of some external medium. The medium is made by this protein. The protein has an inner tunnel. Okay, please see carefully. On the other side, when we will reach the other side, please see the other side. It is hollow. You can see this, right? You can see this hollow tunnel. Yeah, here. So you can see this hollow tunnel. What I am trying to show you here, this hollow tunnel represents an space through which substance can move. Okay, and only substances which have a diameter less than this tunnel, they can only pass, right? Suppose you have this classroom, right? In your classroom, khali cannot come. If I invite, I can see the size of your classroom door, okay? Or you cannot invite a sumo, a sumo wrestler into your room, or maybe Triple H or John Cena. You cannot invite into your room and they can enter. They cannot enter without bending. Okay, they will have to squeeze themselves out in your door. Or suppose we make your door a bit smaller also, make it a window. So because you people are slim, because you people are small students, so yeah, everybody, you are all children. So you can basically, if there was a window in your room only for entering, you can enter. But a WWE wrestler like John Cena or Rock or somebody else, they cannot enter because their body size is too big. Okay, so similar is the case with facilitated diffusion. Substance will be transported across a protein. So protein is not expending energy. Protein is not expending energy. Okay, this is the thing. Protein is not spending energy at all. Okay, without spending the energy, it is allowing the substances to move across it. This is what facilitated diffusion is all about. Without expending energy, the protein is basically allowing the things to come. So how it is happening? It is happening because a substance is not moving against its concentration gradient. Wherever one thing is larger in concentration, it has a natural tendency to move in that direction in which the concentration is lesser, from high to low. All substances have a tendency to move from high to low concentration. This is their natural tendency. If you suppose you have a, like, if you have a perfume bottle, why does the perfume escape outside? Because inside the pressure is more. Inside the pressure is very high. As soon as you open the nozzle, the volatile gases move from one end to other end, and eventually they will spread in the whole room. Okay, so this is the tendency of substances to move from anywhere where their concentration is high to move in that direction which their concentration is lesser. That is what facilitated diffusion is about. So energy will not be spent because it is a natural tendency. So it only allows the molecule which do not exceed the size of the hole. Exactly. It only allows molecules which do not exceed the size of the hole or the tunnel. It's not actually a hole, it's a tunnel basically. Hole is two-dimensional. Hole is present in wall. But this is a three-dimensional structure. We will call it a tunnel. You can call it a hole also, no problem. Okay. So basically a hole or tunnel, wherever the diameter of the hole is lesser, if the diameter of hole is lesser, substance will not pass. If the diameter of the hole is larger, it can allow it to pass. Okay. So this is about the facilitated diffusion. Alright. Okay, so let's write a few things more. You can watch the end of the video. Alright. Yeah, this is another experiment I will show you and I will explain to you, osmosis. For that I have kept this experiment, the rubber egg experiment. I will show you later. Okay. Along the concentration gradient, yeah. One more things a few things more we will write. Okay. Next, please write ATP is not expended, you already know. The rate of transport is dependent, uh, can also be limited, if all protein transporters, are saturated. If they are saturated, then the rate of transport, it will be limited. Okay. Next you can write facilitated diffusion is also very specific. Facilitated diffusion is very specific. It cannot allow everything. It is very specific. It allows substances to, it allows only selected substances for uptake. Uptake means entering. Also, it can be inhibited, it can be stopped, inhibited, or it can be stopped by inhibitors. What is an inhibitor? Can you guess what is an inhibitor? Inhibitors are poisons. Poisons are inhibitors. Spider venom, scorpion venom, 2,4-dinitrophenol, derivatives of nitrophenol, cyanide, all these substances, they are basically poisons, and they inhibit one or more transporter or protein or energy producing machinery protein. They have the tendency to inhibit substances. Okay, that is the thing. Right. So this is about facilitated diffusion. Now there are some other facilitated types of diffusion also. There are types of facilitated diffusion also: symport, antiport, uniport. I will discuss that also. Now there is an important question that has been asked in NEET examination. An important question asked in the NEET examination. The question was, what is the number of units of aquaporin? Aquaporin is a type of porin. Porins have been asked recently in the examination. Okay. Porins are proteins just a second. Porins are proteins which form large pores, in the outer membrane of plastids, mitochondria, and some bacteria. This question has been asked as it is, so note it carefully. Mitochondria and bacteria. bacteria which allow some molecules such as small proteins to pass through. Look, protein is a very large molecule normally. So if protein has to pass through a pore, the pore must be very, very large. Normally proteins are not allowed to pass through by the cell membrane. Protein is a very heavy molecule, but I am telling there are some substances like porins, they can have this structure, they can allow the proteins to pass through. This is a very remarkable thing, such as small proteins to pass through. Small proteins to pass through. Okay. And then there is another question that has been asked in this regard. What is aquaporin? Aquaporin. Maybe it is related to Aquaman. Have you watched Aquaman? Yeah, it's a new movie by Jason Momoa. Not new anymore. I'm sure you must have watched it. Anyway, aquaporin. What is aquaporin? Yeah, okay, you have watched it. Yeah, Jason Momoa is a smart fellow. He looks great and has a great physique. I like him more than the other actors at present. So, what is aquaporin? Actually, it's a water channel. It is a unit of water channel. So, this is the answer you will ask in this question has been asked in your boards. This is important for your boards. What is aquaporin? Aquaporin is a unit of water channel which allows water to pass through, and water channels are made up of 8 types of aquaporins or 8 units of aquaporin. So, please write water channel is made up of water channel are made up of 8 different types of aquaporins. Eight different types of aquaporins. Okay. Now next, right, types of facilitated diffusion. Types of facilitated diffusion. First is uniport, symport, antiport. Facilitated diffusion has 3 different types. So let us discuss these first. Uniport. In this type of transport, only one substance can pass in one direction. So suppose this is a cell membrane. Let me take something else. Suppose this is a cell membrane. And then there is a carrier protein. this. Like this. Okay, wait a second. And now it's better. Like this. Now it is going to allow substance A through this. This is called uniport. This is called uniport. Please write about it. Only one substance, only one molecule or substance moves across the membrane, moves across the membrane, moves across the membrane. Okay, independent of other molecules. Independent of other molecules. So it is not affected by other molecules. It is called uniport. Next, symport. Symport. So it is like two friends going together. Either both will go or nobody will go. Okay. Are there any such friends in your class? Raise your hands. Okay. Only two people will go or nobody else will go. So raise your hands. Who are such friends? Nobody. Okay. Maybe you do not want to raise your hands. Okay, I can see. Yeah, okay. Two people. Yeah. Okay, I saw you. Alright. One more. But you don't have a partner. You are raising your hands by your own. Okay. Alright. I can understand. Okay. So symport is a type of transport in which substances will move only when they are together. This protein channel will allow substances to move only when there are two substances they are present as together. Only then the protein will accept the molecules. Otherwise, it will not allow them to enter individually. So let's take an example of this. It will be like this. This is the protein channel. I am drawing the diameter as slightly larger than last time because now two substances have to move together. So slightly larger than last time. It is only representative. Both substances A plus B, they will move together and they will be exited across the other side of the membrane as such. Okay, this is symport. So please write in symport. In symport, both substances both substances move across the membrane both substances to first of all we should write that there are two substances of different types, two substances or molecules of different types, two substances or molecules of different type cross the membrane through protein transporter at same time, through protein transporter at same time. At the same time. This is called symport. Okay. Next is the antiport. Third option is antiport. So what is antiport? When there are two substances, suppose there are two enemies in your class and they cannot sit together. So when one is entering the class, the other will want to leave, or they will sit at the opposite end or the other side of the row, right? So I am sure there are no enemies among you like these people. So antiport, please write, when two molecules cross the membrane at the same direction, same time in opposite directions. The same time in opposite directions, it is called antiport. Okay. Like this. Like this. Like this. Just a second. I must draw it again. So this is a protein channel. You can draw it any way you like. It just has to be representative. So when substance A is crossing in one direction, the substance B is moving in opposite direction. So please write, in antiport, two different types of substances cross the membrane in opposite direction at same time. Substances cross the membrane in opposite directions, at same time. This is called antiport. Okay. So this is about diffusion. That is all you need to know about diffusion. In your NCERT, there is a chart of comparison between simple diffusion, facilitated diffusion, and active transport. Okay. There is a table. Now you will leave some space table 11.1. Okay. I think it is just after active transport. You will have to just draw this table. This is a table of comparison. It's not a very long table, but I prefer it you draw it by yourself. Just a second. Yeah. So what you have to do is draw a table like this. It's given in NCERT. The first thing we write here, it is property. Then there is a second column of simple diffusion, then there is facilitated diffusion, and then there is active transport. Active transport. Active transport. Okay. So this is if you want to write me to write this down, let me do it for you. Okay. Requires spatial membrane proteins. Spatial membrane proteins. Simple diffusion, no. Facilitated diffusion, yes. Active transport, yes. Highly selective, yes or no. Simple diffusion is not selective. It can select anything. It only needs a concentration gradient. No. It is selective, it is selective. Okay. Transport saturation. Transport saturation. There is no saturation in simple diffusion. There is no saturation in simple diffusion. But in facilitated diffusion, there is saturation. Active transport, there is saturation. Why there is saturation? Because saturation will happen because there are proteins and the number of proteins is limited. If all the proteins are occupied, then that is called saturation. If all the proteins are occupied, there is a limit. Saturation means limit. After some time, if there is excess of raw material to be transported, all the proteins will become occupied. So they cannot transport anymore. But simple diffusion is not selective. You can transport as many substances at many rates possible, but they can be multiple diffusion of many substances from same area. So it is not saturated. Every other means of transport, it is saturated. Against transport, against concentration gradient, it is called uphill transport. Uphill transport means against concentration gradient. Against concentration gradient. Yes or no? You say it cannot be possible. It cannot be possible. It is possible. Okay. Next is ATP requirement. ATP requirement. ATP is not required for this. ATP is required not for this, but only for this. This is basically given in your NCERT. Okay. So this is the active transport versus facilitated diffusion. Now I must discuss active transport. So we have covered simple diffusion, we have covered facilitated diffusion. Now we must cover part C, Now we must cover part C, which is active transport. Active transport. Okay. What is active transport? You can say it uses energy. It uses energy to transport and pump molecules against their concentration gradient. Against the concentration gradient. Against the concentration gradient. It requires specific membrane proteins. It requires specific membrane proteins for this function. Specific membrane proteins. Alright, so it requires specific membrane proteins. Right. So one more thing, because it has requirement of specific membrane proteins, this means that if we are using one type of protein for transporting one type of substance, the same protein cannot transport any other substance. It is so much specific. You cannot use sodium potassium transporter to transport chloride or to transport bicarbonate ions, things like this. Okay, so specific has a specific meaning. The meaning of word specific is that you cannot use one transporter for in place of other transporter. You cannot use one membrane protein in place of another membrane protein for transporting any kind of substance. Okay. They can undergo saturation. They can undergo saturation. They can undergo saturation. Can be inhibited. Can be inhibited by inhibitors. Can be inhibited by inhibitors. Can be regulated by hormones. Can be regulated by hormones. Okay. So this is all about active transport. This is all about active transport. Now we have finished osmosis. Sorry, not osmosis. Osmosis we have to do now. So up till now, whatever substances we are transporting, these are basically solid substances or ions. Okay. Diffusion can be of gases like carbon dioxide, ammonia, any other gas that is produced within the cell atmosphere or by the cells or associated areas. So diffusion can be happening for gases, but for solids, solutes, we need to use facilitated diffusion or we need to use active transport. Now I am going to tell you how solvent is going to be transported. Solvent means, for example, a glass of water in which I place some sugar. Okay. So sugar becomes solvent, sorry, sugar becomes solute and water becomes solvent. Do you understand? Water becomes solvent and sugar is solute. How do you decide which is solute, which is solvent? Do you know the concept in chemistry? Do you know the concept? How do you decide whether something is solute or whether something is solvent? Any idea? How do you decide? What is the criterion? No idea? Say yes or no. Okay. If something is present in less concentration, it is called solute. If something is present in higher concentration, it is called solvent. This is the difference between solute and solvent. Sugar solution, water is higher concentration, you call it water is solvent, sugar is solute. That is the thing. Now I'm going to put the heading plant water relations. By plant water relations, I mean that I want to study how solvent molecules are moving from one place to the other. So basically, I will talk about osmosis, I will talk about water potential, lot of things. Okay. So please write about it. Plant water relations. Water is, all living organisms are made up of majority of water. All living organisms are made up of water to a large extent. Okay. Water to a large extent. Now, why is water important? Because water is the medium in which all the substances, whether sugar, salts, minerals, everything will be dissolved in water. Okay. So please write. All solutes, all substances in cells are basically dissolved in water. Are dissolved in water. Okay. So we need to understand the importance of water, how water is moving within the cell, how water transport is affected from one cell to the other cell. Okay. So we have to study all this. Okay. Let's put the heading water potential. Water potential. Why do we study water potential? To understand the importance of plant water relations. So please put the heading. To be able to understand plant water relations, we need to understand the terms related to movement of terms related to water potential. Okay. So why are we studying this? Because we need to understand the relationship between plants and water. So now let me give you an example what water potential is all about. Look, have you studied electrochemistry yet? In chemistry, has electrochemistry been completed? Yes or no? Please don't sleep right away. I will give you time to sleep after this class. Okay? I understand you feeling sleepy. I think, just a second, I'll show you. 1 second. Okay. Look, yeah, 1 second. I have 3 kinds of solutions. Okay. One is hypertonic, one is isotonic, one is hypotonic. Okay. To understand this, if I keep a cell in a hypertonic solution, that is a concentrated sugar solution, the cell volume will shrink in size. The cell will suddenly shrink in size. If I keep it in isotonic solution, it will remain unchanged in cell volume. If I keep it in hypotonic solution, it will become very much, it will become a burst. It will be immediately, not immediately, but it will somewhere later or sooner, it will burst. Okay. So to demonstrate this, first of all, it is basically osmosis only. I can use an egg. Okay. So we need to prepare an egg in such a manner. So give me a second. I want to show you something. Yeah. Okay. So watch this. This is vinegar. Vinegar is available in our homes, and we take an egg. Okay. And I put vinegar in the egg. Okay. So you have not done this yet, but you should do it definitely sooner or later. So after 2 days, the egg shell has been dissolved. Okay. Now take this egg shell and you can neatly separate it. Watch it. Actually the egg shell has dissolved completely. Only somewhere it is remaining. When it is remaining, it's okay, it's fine. Clean it with a cloth, and after this you get a very soft bouncy egg. It's like a rubber egg. Okay. And you should try this, you will love it. And there are a lot of things we can do. You have highlighters, the fluorescent ink highlighters, the green and the blue highlighters. If I use that green highlighter and place this in dark light is a form of black light, please see what will happen. It's very soft. Maybe you want to touch it, right? It's so soft, right? Okay. Now let's play with it for a while, and let's see what we can do with it. I have not put any colors in it. Okay. So this is yeah, you can put various color lights, LEDs, green, blue, and dark light also, and you will get this kind of thing that you are seeing here right now. I think there is some error in the video. Yeah, okay. Now let's cut it. Okay. nothing has changed inside. Nothing has changed inside. See, it's just like a normal egg. So this egg shell membrane, this is the egg's shell membrane that I am talking about. So everybody should see this by themselves. Take some vinegar, take an egg, and after 2 days, maybe in the next class when we meet, you can have this bouncy egg. You can show me this egg in the next class, okay? So what I am trying to show you here, if I take this bouncy egg and I put it in concentrated salt solution, it will become this, it will look like this. If I take this bouncy egg, okay, if I am taking this egg and I am putting it in hypertonic solution, okay, hypertonic solution, concentrated salt solution or concentrated sugar solution, it will shrink in size, okay, and if I place it in water, it will start, it will show an activity like water will enter inside, so it will become normal shape and size. If I put it in pure water for large amount of time, it might burst after a sufficient amount of time has passed. It will absorb so much water, it can try to burst also. So that is the turgid condition. But these conditions do not happen with plant cell, because plant cell has cell wall around itself. So if it has cell wall around itself, it cannot burst. If it has cell wall around itself, it cannot burst, right? Because the cell wall will prevent the cell from bursting. Okay. So let's write a bit about plant water relations first. Plant water relations first, then we will move on. Water potential. So what is water potential? Water potential is the ability to measure the potential energy of, or the energy of water molecules. Okay. So please write water potential expresses water potential expresses the relative energy of water molecules compared to water molecules of solution compared to pure water. Time up, sir. Okay. Alright, so just write it up and then I'll leave you. Compared to pure water. Okay. So in the next class I'll show you how this is happening. So we'll watch some videos, and do this quickly as we can. Okay. So in the next class we'll take this up. Okay. So thank you, dear students. In the next class we'll finish it. Try to finish this chapter in the next class. Let's see how much we are able to do. Okay. Thank you. Thank you for your time. See you tomorrow.